So imagine you have a highly detailed instruction manual for this impossibly complex machine, let's just say, the human body.
Right.
And now imagine realizing that this manual, it actually changes depending on which room you're standing in when you try to read it.
Which is already crazy to think about.
Right. And it gets weirder because the actual spelling of the words in the manual changes based on, exactly who is reading it.
Exactly.
So that shifting dynamic instruction manual is actually the reality of human biology. And today we're doing a deep dive into a groundbreaking 2026 paper from the journal Cell.
By Cohen and colleagues, yeah.
Yeah, and they finally figured out how to read this manual. They built this tool, it's got a long name, RT&T-AMP-MERFISH, that lets us look at like 33,000 different genetic instructions in the brain.
And it lets us pinpoint exactly what room they're being read in, down to the tiniest individual variations. It's really a massive leap forward.
Because for a long time, we've kind of treated the genome like a static, unchanging book, right?
We have, yeah. I mean, we already know a lot about the genetic code itself. We can sequence a genome very effectively.
Yeah.
But seeing exactly where those genes are actively being used inside, like a three-dimensional slice of brain tissue, and which specific versions of those genes are playing out in real time, that has been just a massive biological blind spot.
Okay, so let's unpack this blind spot before we get to the new tech. Why is this so hard? I feel like we sequence things all the time.
We do, yeah.
Why can't we just take the brain tissue, sequence it like we usually do, or, I don't know, just put it under one of our wildly powerful modern microscopes?
Well, that is the core spatial dilemma right there.
Yeah.
Think about standard bulk RNA sequencing. To do it, you basically have to take a piece of tissue and grind it up into a cellular smoothie.
The smoothie, okay.
Yeah, and when you sequence that smoothie, you get a fantastic, highly accurate list of all the genetic ingredients that were active. But you completely lose the zip code.
Right, a specific RNA message was in the tissue somewhere, but you have, no idea which exact cell produced it or who its neighbors were.
Exactly. You lose the spatial context entirely. Now, to address your second point about microscopes, we do have spatial technology like standard FES.
Fluorescence in situ hybridization.
Right, which essentially uses these tiny probes to make specific RNA glow inside the intact tissue. But traditional imaging has a really severe limitation when it comes to short RNA sequences.
Why is that?
Well, to get a bright enough signal to actually see it under a microscope, standard FES requires you to attach dozens of fluorescent probes to a single piece of RNA.
I'm stuck on something here, though. If an RNA transcript is short, or if we're looking for tiny, tiny variations in the code, why not just use smaller probes?
It's not about the size of the probe. It's about the physical space on the target RNA. You simply can't fit enough probes onto a short sequence to generate enough light for the microscope sensors.
Oh, I see.
Yeah, if you try to force it, the signals just overlap, and you get what we call signal crowding.
Signal crowding?
Yeah, it just looks like a glowing blur.
So if you grind it up, you lose the location. If you look at it whole, it's too blurry.
Exactly.
Enter the first half of our massive acronym for today. RT&T-AMP.
Reverse transcription and transcription amplification.
Right. So I want to know exactly how this solves the blur problem. Don't gloss over the chemistry here. Let's really get into it.
Oh, I won't. Let's trace the mechanism end to end because it's brilliant. Step 1 is fixation. The scientists use a glyoxyl-based fixative on the brain tissue.
Like a glue.
Think of it as a highly specific molecular superglue, yeah. It permeates the cells and locks all the fragile RNA molecules exactly in place, anchored to the cellular matrix so they cannot drift away.
Okay, so the RNA is frozen in space. What's next?
Step 2 is reverse transcription. They introduce a poly-T primer into the tissue.
Poly-T, okay.
Now, natural messenger RNA has a tail made of a long string of adenine bases, a poly-A tail. The poly-T primer is perfectly complementary, so it seeks out and binds specifically to that tail.
Like Velcro.
Exactly. Then they send in an enzyme called reverse transcriptase, which reads the RNA and builds a complementary DNA strand, a cDNA.
Okay, make a copy.
Right. But here's the critical mechanism. When the enzyme reaches the end of the RNA, it has a quirk. It naturally adds a tiny non-templated overhang of 3 cytosine bases, a CCC sequence right to the end of the new DNA strand.
Wait, why does it add 3 cytosines? Is that like a mistake by the enzyme?
It's an inherent biochemical feature of the enzyme, basically a byproduct of how it falls off the strand. But rather than fighting it, the researchers exploit it.
Oh, smart.
Which brings us to step 3, template switching. They add a template-switching oligo or TSO.
Okay.
This TSO has a GGG sequence that perfectly matches that random CCC overhang. It snaps together like a molecular handshake.
So the enzyme leaves a little CCC tail, and we send in a GGG puzzle piece to click right into it.
Precisely. But here's why that handshake matters. This TSO carries a very specific piece of extra code called a T7 promoter sequence.
Okay, a promoter sequence.
Yeah, so the reverse transcriptase enzyme grabs onto this molecular handshake, switches tracks onto the TSO, and just continues copying. It effectively welds the T7 promoter onto the end of our new cDNA strand.
So let me make sure I'm visualizing this perfectly. We took the original RNA, superglued it to the cell, made a DNA copy of it right there on the spot, and then cleverly used the enzyme's own quirk to paste a brand new T7 promoter onto the end of that copy.
You nailed it.
What does the T7 promoter actually do, though?
That is step 4, transcription amplification. The T7 promoter is essentially a flashing neon start sign for a different enzyme called T7 RNA polymerase.
A start sign.
Right. When you wash that enzyme over the tissue, it binds to the promoter and starts churning out dozens, sometimes hundreds, of new RNA copies from that single DNA template.
Wow.
It creates a localized pile of RNA called an amplicon. And because the original molecule was superglued in place, this whole pile of new copies is sitting in the exact physical spot where the original single transcript lived.
That is wild.
Finally, they use paraformaldehyde to lock all these new copies in place so the pile doesn't diffuse.
That is incredible. I'm thinking of it like a library copier.
A library copier.
Yeah, like the old way, grinding up the tissue is like taking all the books out of a massive library in a giant pile just to read them.
Right.
You know what the books say, but you have no idea which specific shelf they came from.
Exactly.
And standard imaging is like standing in the dark library, squinting at tiny font you can barely see. But RT&T-AMP, it's like installing a magical 3D photocopier right on the specific bookshelf.
That's a great way to put it.
It prints out bright, glowing copies of the exact tiny sentence you need, leaving them stacked right there on the shelf so it's absolutely impossible to miss them, even in the dark.
That is a perfect synthesis of the mechanism. By creating that amplicon, you amplify the physical target at the exact site of origin, which means you now only need a tiny handful of probes, sometimes just one, to clearly see the RNA under the microscope.
Totally bypassing the signal crowding process.
Bypassing it entirely, yeah.
Okay, so think about your own field of work for a second, to everyone listening. How often does having the context or the exact location of a piece of data completely change its meaning? It's huge.
It changes everything.
Right. So we're about 5 minutes into this deep dive. Let's do a really quick bullet point recap of what we just covered.
Good idea.
Number 1, the spatial dilemma. Grinding up tissue loses the location, and standard microscopes are too blurry for tiny RNA.
Number 2, the RT&T-AMP solution. We use fixatives to glue the RNA in place and a quirky enzyme to build a DNA copy with a special T7 promoter tag.
And number 3, that tag acts as a start sign to put a massive pile of new glowing RNA copies, an amplicon right on the specific bookshelf where the original RNA lived.
Perfect recap.
Right, so printing a giant pile of copies for one gene makes total sense. But we mentioned at the top that they map 33,000 distinct RNAs.
We did, yeah.
That's 23,000 genes and 10,000 specific variations. If you print glowing piles for 33,000 different things in a single cell, doesn't the entire cell just turn into a blinding white light?
You would think so, yeah.
How on earth do they scale this up without it descending into total chaos?
Well, this is where the second half of our acronym comes in, MERFISH. It stands for multiplexed error-robust fluorescence in situ hybridization.
Another mouthful.
Right. Because we successfully amplified the targets, we can now use the MERFISH protocol to read them systematically. They don't just assign one color to one gene. They use a 90-bit barcode system.
90 bits. So we're talking about treating the brain tissue like a massive digital hard drive.
Exactly.
Reading a string of 90 ones and zeros for every single gene.
Yes, and they read those bits over the course of 60 separate rounds of imaging.
60 rounds.
60 rounds. So during round 1, they wash a specific set of fluorescent probes over the tissue. If a probe binds to an amplicon, the microscope reads a 1 for that target in that round.
Okay.
If it doesn't bind, it reads a 0.
But wait, if they do this 60 times, won't the probes just pile up? How do they read round 2 without the light from round 1 interfering?
That's where a highly precise chemical trick comes in. The fluorescent dyes attached to these probes are TCEP cleavable. TCEP is a chemical reducing agent. After the microscope takes its picture for round 1, they wash TCEP over the tissue. It acts like highly specific molecular scissors.
Oh, scissors.
Yeah, targeting and snipping the disulfide bonds that hold the glowing dye to the probe. The dye washes away, leaving the tissue completely dark again.
Oh, that's wild. So it doesn't destroy the RNA we amplified.
Not at all.
It just neatly clips the light bulb off the wire.
Exactly. It's a form of click chemistry. You wash the probes in, take the picture, clip the light bulbs off, wash it clean, and start round 2.
That is so elegant.
And over 60 rounds of this flashing and erasing, the unique chronological pattern of on and off signals builds a distinct 90-bit barcode for every single one of those 33,000 RNAs.
Wow.
And the code is mathematically error-robust, meaning even if the microscope accidentally misses a single flash of light in, say, round 42, the overall 90-bit code is complex enough to self-correct.
So it still accurately identifies the gene. That is a staggering level of engineering. So they have this God mode view of the tissue now. What do they actually see when they turn it on?
Well, there are so many discoveries, but let's look at smooth muscle cells.
Oh, right. The paper has this fascinating real-world biological case study about SMCs that completely changes how I think about cellular identity.
Yeah, the smooth muscle cell data is a perfect illustration of why spatial context is absolutely non-negotiable. SMCs are the cells that wrap around our blood vessels to help regulate blood flow.
Right.
Traditionally, we just thought of an SMC as an SMC. But when the researchers mapped their gene expression in 3D space, they found that these cells completely alter their biological identity based purely on how crowded they are.
The physical density actually changes the instruction manual they read from.
Exactly.
Because when the smooth muscle cells are densely packed together, like hugging tightly around a vessel, they express contractile genes like ACT2 and MY11.
Right, their whole world is focused on squeezing the blood vessel.
But when they are sparsely distributed, when they don't have many neighbors, they shift their entire genetic state. They start reading different genes, vascular remodeling genes like CXCL12 and EGAL7.
And if we look at the implications of that, it proves that a cell's microenvironment literally dictates its function. You would never, ever know that if you just ground up the tissue in a blender.
Yeah, of course not.
You'd see a confusing mix of contractile and remodeling genes, and you might assume you have 2 entirely different subtypes of cells, rather than one type of cell simply reacting to its spatial zip code.
Think about what this means for your own body. To our listeners, if your cells are completely changing their core identities just based on how crowded their local neighborhood is, how does that change the way you think about tissue health?
It's a huge paradigm shift.
A disease might not always be a fundamentally broken cell, right?
Okay.
It might just be a perfectly fine cell reacting to a break in neighborhood.
Exactly.
And it's not just about crowding, is it? It's about who they are actively talking to. Because the paper also dives deep into mapping cell-cell communication.
Right. They mapped ligand receptor pairs because they have the exact 3D coordinates of every single cell, and they know exactly which genes those cells are expressing. They can physically listen to the cells whispering chemical signals to each other.
Whispering?
Yeah. A ligand is the molecular signal, like a key, and the receptor is the lock on the receiving cell. For instance, they mapped the PDGFB ligand, which is produced by endothelial cells that line the inner walls of blood vessels.
And who are the endothelial cells talking to?
They saw that ligand physically co-localizing in space with the PDGFRB receptor on adjacent pericytes and astrocytes. This specific conversation is vital for maintaining the blood-brain barrier.
Wow.
They also mapped Notch1 signaling across different cell types, and perhaps most fascinatingly, they mapped the dynorphin ligand.
That's a natural opioid peptide, right?
Yes, they mapped it communicating with the kappa opioid receptor.
So they're basically watching the brain's internal pain and regulation network light up in native 3D space.
Live chatter right there on the map.
Okay, so we're hitting that 10-minute mark. Let's do our second quick bullet point recap for the listeners.
All right.
Number 1, they scale up using MERFISH, which relies on a 90-bit barcode system over 60 rounds of imaging.
Number 2, they use TCEP click chemistry to clip off the glowing dyes between rounds so they don't get a blurry mess.
Number 3, spatial mapping showed us that smooth muscle cells completely shift their gene expression based just on how crowded their neighborhood is.
And number 4, We can literally map ligand-receptor pairs to eavesdrop on cells talking to each other, like the dynorphin pathway.
Eavesdropping on cells. I love that.
Mm.
Okay, that brings us to what might be the most revolutionary part of this paper. We've talked about genes, but we really need to talk about isoforms.
Yes, isoforms are the big frontier here.
I know the word isoform gets thrown around a lot in genetics, but let's be precise here. If a gene is the master instruction manual, how does an isoform actually happen?
Okay, think of a gene as a master recipe for a cake. The genome has the basic, unedited instructions, but our cells have a regulatory process called alternative splicing.
Alternative splicing.
And they can also use alternative transcription start sites. This essentially means the cell's machinery can edit the master recipe on the fly as it reads it.
Okay.
It can splice in a sequence for chocolate chips, it can swap out regular sugar for honey, or it can skip the frosting sequence entirely.
So the resulting protein variations are the isoforms.
Yes, they originate from the exact same master gene, but they have slightly different sequences and can have vastly different functions in the body.
So the gene is the base recipe. The isoform is the specific variation that actually gets baked in that specific room of the brain.
That's it.
But wait, if these isoforms are just tiny variations of the same gene, isn't that incredibly hard to spot?
It is incredibly hard. The differences between two isoforms might only be a few dozen base pairs, tiny snippets of genetic code. Previous spatial technologies completely missed them because they couldn't get a clear enough signal on those tiny, unique regions. They couldn't tell the chocolate chip cake from the raisin cake. But because RT&T-AMP amplifies the signal perfectly in place, the researchers managed to map 10,000 distinct isoforms. And they did it using just 4 to 6 specific encoding probes for each isoform.
That's amazing. Let's look at the biological case studies here because this is where the implications are just mind-bending to me. Let's start with the MEF2C gene.
Okay, MEF2C. It's a gene that is absolutely vital for learning, memory, and neuronal development.
Crucial stuff.
Very. When they mapped it, the researchers found that different physical layers of the cortex use entirely different spellings of this exact same gene.
Really?
Yes. Isoform 226 is heavily expressed deep down in the lower layers of the cortex. But isoform 218 is pushed up into the upper superficial layers.
The same base gene, different variation, strictly segregated by physical depth.
Exactly.
That is wild. But there's an even more extreme example in the hippocampus, right?
Yeah.
The memory center of the brain. They looked at the TAFA1 gene.
Yes, the TAFA1 example is fascinating.
Because the paper shows that isoform 202 of this gene is spread out broadly across 3 different regions of the hippocampus, the CA1, CA2, and CA3.
Right.
But isoform 201 is strictly confined to just the CA1 region. It is totally absent right next door in CA2.
And this raises a profound biological question, because here is the twist. The actual final protein that both of those TAFA1 isoforms produce is 100% identical.
Wait, hold on. Identical.
Identical. The difference between isoform 201 and 202 is only found in their untranslated regions, or UTRs.
If the final memory protein that gets baked is exactly the same, why does the brain go through all the trouble of making a different isoform for it just because it's in a different part of the hippocampus?
Because the untranslated regions aren't the recipe for the protein itself. They act as the logistical shipping and handling manual for that protein.
Oh, a shipping manual.
Yes. The UTR tells the cell when to build the protein, how fast to synthesize it, and exactly where to deploy it inside the cell's architecture.
Wow.
So the brain is specifically regulating the logistics of this memory-related protein depending on its exact millimeter zip code. It wants the protein managed and deployed differently in CA1 than it does in CA2.
That makes so much sense. It's like a car company making the exact same engine part in 2 factories. But the CA1 factory gets a shipping manual that says, rush delivery, put it on the front line immediately.
Right.
And the CA2 factory gets a manual that says standard delivery, keep it in storage until needed. We would never see those shipping labels if we just ground up the tissue to see what engines were made.
That's a really great way to think about it. And they found an even more specialized example in a region called the choroid plexus.
That's the blood cerebrospinal fluid barrier, right?
Yeah.
Basically, the brain's heavy-duty filtration system.
Yes. The choroid plexus is wildly rich in specific isoforms. They looked at a gene called SLC301A1.
SLC31A1.
Yeah, it encodes a copper transporter. And copper regulation is vital for brain metabolism. They found that isoform 202 of this gene is expressed pretty much everywhere in the brain. It's the standard copper transporter.
Okay.
But isoform 204, it is found exclusively in the choroid plexus and nowhere else. The brain's filtration system demands a highly specific version of that transporter that the rest of the brain simply doesn't use.
Amazing. Okay, so before we wrap up, let's do our 15-minute recap bullet points.
Sounds good.
Number 1, isoforms are specific variations of a base gene created by alternative splicing or start sites like tweaking a cake recipe on the fly.
Number 2, RT&T-AMP-MERFISH mapped 10,000 of them because it can pick up tiny sequence differences perfectly in place.
Number 3, the brain uses different isoforms in specific areas, like MEF2C varying by cortical depth.
And number 4, sometimes the protein is identical, but the untranslated regions differ, acting as localized shipping manuals, like we saw with TAFA1 and the choroid plexus transporter.
So to everybody listening, what stands out to you from all of this? Does knowing that your brain uses entirely different spellings of a memory gene, depending on the exact millimeter of tissue, just completely blow your mind? Because it does mine.
It's incredible.
So what does this all mean when we zoom all the way out from the RT&T-AMP enzymes, the 90-bit barcodes, and the click chemistry? What is the grand takeaway from this paper?
Well, the grand takeaway is that the era of simply taking a roll call of genes is over. We can no longer just look at what genetic code is present in a tissue. To truly understand brain function, how tissue stays healthy, and ultimately how disease takes root, we have to look at where those genes are physically located, who those cells are actively whispering to, and exactly which specific version, which isoform, is active in that exact microenvironment. The spatial architecture is fundamentally as important as the genetic code itself.
It's the difference between having a disorganized list of parts for an airplane and actually seeing the spatial blueprint of how they fit together to fly.
Exactly. And if we look forward to the future, the authors present a truly mind-expanding idea in their discussion. We know that the physical structure, that literal 3D folding of DNA, along with epigenetics, influences how these specific isoforms are chosen. Imagine a future very soon where we pair this spatial RNA mapping with spatial epigenomics.
Oh, wow. So we could physically see the localized folding of the DNA inside a single cell that forces it to choose the raisin cake recipe instead of the chocolate chip.
Yes. We could see the structural cause and the resulting RNA effect simultaneously in intact tissue. Or, even more radically, think about therapies. With precision tools like CRISPR-Cas13, which can target RNA, we wouldn't just be blindly turning a gene off everywhere in the brain.
Right, because that could cause awful side effects.
Exactly. If we know the exact spatial zip code of a diseased isoform, we could go in and edit just one specific isoform in one specific millimeter of the brain to treat a localized disease, leaving the rest of the brain's completely normal recipes perfectly intact.
That is surgical precision on a molecular 3D scale. It's unbelievable. So as we wrap up today, I want you listening to really mull that over. Think about how this high-definition, spatially aware view of biology might completely revolutionize the medicine of tomorrow.
It's a whole new world.
We are no longer just reading the instruction manual for the human body. We are finally learning how to read the room. Thank you for joining us on this deep dive.